One of the other issues that we deal with is how do we look at this tremendous diversity? And we've heard all the facts and figures about the billions of microbes and the kilometers of hyphae that we see in the soil. But it's still a very big challenge to deal with this. And so I'm going to talk a little bit about some of the methods that we use to address and measure and monitor some of these microbial communities. And so we can look at these at one level and talk about the whole microbial community. It's more of an approach of saying, I don't really care what their names are. I'm not so fussed about their identity, but I'm interested in patterns of change in those microbial communities. And so we've got a range of techniques that we use for that, and we often try and relate that back to functions like nutrient cycling or carbon sequestration that we're interested in. The other approach that we can use is to look at functional groups. So these are groups of organisms, so we heard about the mycorrhizal fungi, for example, today, where we know who they are and we know what they do. And so what we can do there is start to look at them and identify them and manipulate them and do experiments where we can take them away and add them back into the soil and get an understanding of their functioning. And so again, what I'm interested in is how do we span these two things, going from the whole community where you have your tens of thousands of different species and looking at specific functional groups as well. Often when you talk about soil biology and soil ecology and its management, people talk about these concepts of resistance and resilience, and there's often a lot of confusion about what these terms mean. And so I just want to talk a little bit about the terminology. So if we imagine that we have a period of time here, and then we have something that we care about on our y-axis, doesn't matter what it is, it could be plant growth, nutrient content, there might be some stress, and we can see a decrease in the growth of that plant, for example. So we've had this perturbation, this stress that's had a negative impact on our system. If we have a resistant system, what will happen is when we get our perturbation, it resists that stress and it continues operating in the way that we want it to. So that's the concept of resistance. It's able to resist a stress or a perturbation. Resilience, on the other hand, is when a system is challenged with a stress, you might see it go down, but then it bounces back. It's resilient. And so what we need to build in our soil systems is both resistance and resilience. We want to be able to resist the challenges and we want to be able to bounce back. Okay, so resistance and resilience, I think, are key targets for management of soil ecological systems, and indeed any system. We could be talking to a room full of economists and you would want a resistant and a resilient economy. Okay, so it's those same principles that apply there. Okay, so soil biology, lots of exciting things happening in front of me that you can't see. Okay, so soil biology, to me, and I think this point was made by Walter as well, is there are no silver bullets. That's my personal opinion. There's not one magic microbe that's going to come along and save the day. It's about managing the microbial communities that you have to try and bolster them to build that resilience and resistance in your system. That should always be the first port of call. So building that resilience and resistance. The soil is a tremendously difficult place to live. I think it's really interesting to think about the environment that microbes and organisms exist in when they're in the soil. If you took this barn and treated it as the soil, we could fill it up with different particles. So golf balls and tennis balls and footballs and beach balls and cars, all these particles of different sizes that range over many orders of magnitude in size. And so we've got the solid material and then we've got the spaces between them. And then suddenly someone turns on the tap and fills the room with water. Okay, suddenly we've gone from a dry environment where we're trying to navigate our way through all these particles to one that's very wet. But we're in a well-drained environment and suddenly that water drains away. And so on very short timescales, the organisms in the soil have to contend with an extremely dynamic environment. It's changing tremendously quickly, and that has a really big impact on their behavior and their function. And so building some stability, and as we heard before about structure in the soil, is important. I think the other thing is, as scientists, we like to be a bit reductionist. We like to say, let's get right down to one particular thing. But what we really need to think about is the complexity of the interactions that we see in our system. It's not about one organism, one group of organisms. It's how they're interacting with all the other processes that take place in their soil. So we need to take a really broad view that embraces that complexity. And we've heard all the statistics before about the numbers of microbes and kilometers of hyphae. I put this slide up, and it tends to upset a lot of plant scientists because as a soil microbial ecologist, I always stand up and say, well, most plants don't have roots, they have mycorrhizas. So mycorrhizas are these normal things that somewhere about 80 to 90% of plants form. They're associations between soil fungi and plant roots. This is what most plants do most of the time. Okay, this is what's normal. The reason I like this slide is because this is the root here, there's another one here and another one here, and all that messy white stuff is the fungal hyphae. Okay, so that's an interface between the soil and the fungus and the plant. So when you think about the soil environment, try and keep this sort of image in mind. There's this tremendous diversity and mass of biomass that we see below ground. Okay, I'm going to give you a bit of a slideshow here, and let's look at some microbes and some organisms in the soil. So this is a nematode, so we've got our nematode here, and it has wrapped itself around a piece of organic matter, a small twig in the soil. So these are some electron micrographs. So you can see a nematode. It gives you a sense of the scale of these organisms. This is a really, I think, a fantastic image. It's one of the best ones I've seen. This is the noose of a nematode trapping fungus. So this is a fungus that grows in the soil and it forms a noose. And when one of these nematodes comes swimming through, it can get entangled in that noose. The fungus then releases enzymes that degrade the nematode and it basically consumes that material. So we're moving nutrients from the body of the dead nematode into the fungus, into the soil biota. This is an image of a mite. So he's foraging on what is a corn cob. So we've got a corn cob here, and then this is the mite. You can see his legs, his head's down here. If we have a bit of a closer look at his mouth parts, I would not want to meet this guy in a dark alley. Okay, and remember, think about the soil environment. You come around the corner and this guy's waiting to meet you with this rather sinister looking device to consume you. Okay, so it's a challenging and tough environment. It's eat or be eaten. If we look at the same mite, we're going to have a look at the back end of the mite here. What's of interest here is we have cells and bacteria on the surface of the mite. Okay, so even the organisms in the soil are an ecosystem for other organisms. So it's all this interconnectedness that's really important. If we have a closer look again here, you can see these bacterial cells in some more detail on the back end of the mite. Of course, everything dies at some point in time, and so here we've got a deceased mite, and it's starting to be entangled by fungal hyphae. So this thing's died, and the fungi are growing on the surface of the mite, and they're degrading it, and they're making use of those resources. Okay, so they're consuming the body of that dead mite. These are some examples of fungal hyphae growing through the soil. So again, we can see the hyphae growing down through here, and we've got some bacteria on the side there as well. This is a soil aggregate. So this is an electron micrograph of a soil aggregate. So you go out into the soil, you get your handful of soil, and you get those little aggregates, maybe a couple of millimeters in size. This is what they look like up close. Okay, so this one here, what I want you to do is take note of that little bump up there. Okay, so we're going to zoom in on that aggregate. So here we've got that same bump here, and if we zoom in on this particular region here, we've got our nematode trapping fungus again. Okay, so it gives you a sense of the scale. If I go back, here's our aggregate. Now you can hopefully see the nematode trapping fungus as we get closer and closer. Okay, so think of those aggregates as really vibrant, large, active microbial communities. They're a really important part of the soil. This is just an example of two aggregates taken from fields on the same soil type, but they've been managed in different ways. So this one's been organically managed for some time, this one's been conventionally managed for some time. I'm not going to go into all the details about the differences. The point is that management has an impact on the nature of the structure of the soil. And so a lot of that's driven by the soil biology. Okay, so that's a bit of a quick whistle-stop tour of sort of who's who in the zoo. What I want to do now is talk a little bit about how do we study soil biology. And the one thing I can say really with any certainty here is there are a vast number of methods and approaches that we can use. Okay? And of course, none of them is going to be the best or the worst. They all have their own strengths and weaknesses, their pros and cons. So for example, we could look at the microbial biomass. How much biomass of microbes are in the soil? So we can measure the microbial biomass carbon. We can measure the total lipid content of the soil. We could look at the extent to which the fungi grow into the roots. They're all measures of the biomass of the soil biota. We could look at the activity of the microbial communities. We could say, don't care who you are, don't care what your name is, but I'd like to know what you do. Okay, so it's getting a sense of what they do. We can look at their respiration, the activity of the enzymes. We can look at the rate of substrate decomposition. So there's a whole host of approaches that we can use to look at the activity of these organisms. We can look at the diversity. So knowing if we have a more or less biodiverse system. So we can look at the abundance of cultural organisms. We can look at the identification of spores of fungi. We can look at the lipid profiles of the soil. We can look at the DNA and the RNA profiles of the soil. And even then, if we subdivide down into the molecular and DNA-based approaches, there are dozens and dozens of different methods that you can use. The point is that this is a very long list of approaches. Okay, and so as I said before, there's no one particular one that's better or worse than the others. People always ask me which one to use, and I'm going to give the really good scientist answer and say it depends. Okay, it depends on the question that you want to ask. Okay, so it's really about which one do I use? Well, it depends on what you want to know. Has my soil got more microbial biomass? Measure the microbial biomass. Is my soil more effective at cycling nitrogen? Measure something about nitrogen cycling. Is there more diversity in my cell? Take some measure of soil microbial diversity. So really, it depends on the question that you want to ask. Second most common question I get is, what do I need for my soil to be healthy? Again, as a good scientist, I'm going to say it depends on the context. Defining soil health is very context dependent. What is a healthy soil? Let's take a sand. What is a healthy sand if you're a sand dune is different from if you're in a cropping soil that's sandy? Okay, so it depends on the context. I can't give you a list of all the organisms that you need in their relative abundance. Okay, but what I can tell you about is how those things change in response to your management. And I think that's a really key point. It's really about how does my system change and is it changing in a positive way? Am I increasing my microbial biomass? Am I increasing my nutrient cycling functions? Am I increasing my diversity and so on? So what I want to do now is just run through quite quickly a number of examples of where we've taken different measures of soil microbial processes or biodiversity or activity and related them back to management. Okay, because I think that's really the important lens to look at this through. So this is just a very simple figure. It shows mycorrhizal colonization of roots. So this was a study we did in Gippsland where we worked with a group of pasture farmers through a land care organisation, and they wanted to know about the impacts of their management on their soil health. One of our measures was mycorrhizal colonization, so these beneficial associations. It varied between soil types and it was impacted by soil fertility. I'm not going to get into the details of that too much, but I want to think about it in a broader sense. So one of the things that we can do as scientists is we can do our experiments and we can measure things in the field, but we also have a tool called a meta-analysis, which is where we can go into the scientific literature and collect all the data around a particular question, and then we can analyze that data to try and find bigger patterns. So hence this term meta-analysis. And so what we wanted to know was in this concept of ecological intensification, so changing management more to one that relies on ecosystem services, could you see an increase in the formation of these beneficial associations, and was that impacted by management decisions? So we looked at field studies that compared different cover cropping and non-cover cropping treatments and conventional versus alternative tillage treatments. So two obvious management practices that we can implement. And then we looked at the formation of these symbioses in the cash crop, the crop that the farmers were interested in. So we collected data from all around the world. We had nearly 50 total studies where people had looked at these alternative tillage and these cropping and cover cropping practices and their impacts on mycorrhizas. To cut a very long story short, what we found was that if you had a cover crop in your system, your mycorrhizal colonization was about 30% higher than if you had a fallow. So maintaining plant cover had a benefit in terms of the mycorrhizal colonization, these beneficial symbioses in the following cash crop. Okay, simple management practice. What about tillage? We found that alternative tillage practices had about 35% higher mycorrhizal colonization than where you had conventional tillage. And this is across a whole host of different farming systems. So we've lumped that all together. But again, a clear impact of management. Now, where I think this gets really interesting is if you think about different farming systems, right? You have to make choices, and it's not for me to tell you which choices are the right choices, but I can say something about their potential impacts. So if, for example, you're an organic farmer, you're not going to use herbicides in your system. So the way you control your weeds is through tillage, okay? That's going to have an adverse effect if you've got this conventional tillage. It's going to knock out these beneficial fungi. But if you put a cover crop back in the system, you can regain that loss back. Okay, so there's a trade-off in the management decisions and interrelatedness. Ideally, you'd be in a system where you had alternative tillage and cover cropping. What you want to avoid is a system where you've got conventional tillage and a bare fallow, if what you're interested in managing is these symbiotic associations. Okay, I'm going to change directions a little bit here and talk about measuring function. Okay, so nitrogen cycle, if you go to Google and type in nitrogen cycle, you'll get a million different versions of this. What it's really about is the organic matter being converted from an organic form into a mineral form, so ammonium and nitrate, that can be taken up by plants. Okay, so it's taking this organic matter, these dead roots, these dead organisms, and the organic material in the soil and releasing nutrients. The first step in this process is mineralization. Okay? And so we can measure mineralization in the lab quite easily through what we call potentially mineralizable N. We take some soil, we incubate it, we measure the amount of ammonium at the beginning and at the end of that incubation under certain conditions, and it tells us something about how quick this process is happening. How rapid is the cycling of the organic matter in the soil occurring? And so again, with that group of farmers in Gippsland, pasture producers in Gippsland, we found quite a bit of variation, and we found it was related to the texture of the soil. Okay? But within those soil types, there's considerable variation. What's this farmer doing that this farmer's not doing? Okay? And so that's where drilling down and looking at those things is really important. The other thing to do is to say, okay, I'm this farmer here, I've got a low rate of N cycling, I spend a lot of money on nitrogen fertilizer each year, I want to boost that up. Brilliant, thank you. I want to increase that rate of that process. We've measured it. Let's go back in a few years' time after they've implemented some different management practices and see if they're moving up in this direction to what's possible. So benchmarking where you are is really useful, and then looking at change through time, I think, is essential when you're looking at soil biology. I can't say the right number's 15.5. Okay, I can't say that. No one can tell you that. But what they can tell you is whether or not things have changed. Okay, this is an example of another study where we were looking at revegetation on farms. Okay, so this was some work in northern Victoria. This was a riparian restoration project, so obviously this is the unrestored system where the livestock were able to get down into the stream. Erosion was a problem, eutrophication was a problem. This was one of our experimental sites, so we revegetated 1 kilometre of stream, so you can see we've put in a fence line, and then all these are the little tree guards around the trees. And so we set that up, and over a period of about 10 years, we looked at change in these systems. As part of that work, we also looked at revegetation projects that have been done over the past 50 years to try and see if there was an increase in soil carbon by revegetating these systems, and could farmers get paid for that? And then also looking at the impacts on the health of the soil. And so the way we looked at this is we looked at the diversity in the biomass of the microbes. And so this was a lipid-based approach, so phospholipid fatty acids is a term you might have heard of. And what this tells us is that over time, so this is years since the trees were planted, we're seeing after about 20 years, we're getting a build-up in the microbial biomass in the soil. So it can take a long time for these things to change, but you can start to detect a change through time. The other thing that we did with these data is we looked at the dissimilarity between the revegetated soil and the other side of the fence line. So we made a comparison. Here's the paddock that we didn't restore, here's the one that we did restore, and we went back and looked at them through time. And what we found is if you compare a pasture and remnant vegetation, so never farmed vegetation, the microbial communities are really different from one another, these red lines here. The further apart they are, the more different they are. If you look at younger sites, so here, this one was restored 9 years ago. It's very similar to the adjacent pasture. If you look at a pasture next to a pasture, they're obviously very similar. The message here is that through time, those communities became more dissimilar. And so that's one approach. If you want to say, what's the health of my soil in this management, you need to compare it to something. You need a benchmark. So you've got two approaches. You either do something and you come back in 20 years' time, or you try and match that paddock to another paddock that's had similar prior management and then look for change there as well. Because sometimes you don't have 20 years to wait for the answer. Okay, so it's about how you go about that sort of approach. Omics, this is a hideous slide, but I really like it. There's many hours that go behind this and many tens of billions of data points in there. This is looking at the entire microbial community in a soil, okay? And so there are thousands and thousands of species in there. And there's lots of changes. There are actually different experimental treatments here. This treatment in particular, obviously a big change here. I'm not going to go into the details of it, but I think you're going to hear a lot more about omics. So omics is really, one of the omics is looking at DNA. It's looking at the microbiome. Okay, you can't open the newspaper without reading some story about how our gut microbiome has an impact on any aspect of our health. The same is true of our soil. Okay, and in fact, I would argue that the soil field in microbiomes is light years ahead of where the medicos are. They've actually suddenly realized that microbes are important. When I went through university, we were told there were no microbes in your gut. Okay, right? There's none in there, it's acid, nothing's going to grow in that environment. And now suddenly they're telling us there's vast amounts of microbes in there. You can buy them at the supermarket even. Okay. But you guys already knew this, right? You've been dealing with microbes in your soil for a long time. And in fact, farmers have some of the healthiest gut microbiomes on the planet, right? Because you're out there in the environment. You're not sitting inside all day in a nicely air-conditioned office. Okay, so I'm nearly at the end here. How do we study soil biology? There is no one best method. Okay, I'm not going to tell you one method's better than the other. I'm going to say it depends on the question you want to answer. What I would say is adequate sampling is really, really important. Go out and get a representative sample of your paddock. Okay, so sample in an adequate manner. Apples and bananas, make sure you're comparing like with like. I've often looked at data sets where farmers sampled this paddock and they've sampled that paddock, and this is the one that's had their management practice, and it's vastly different from the other one. But when you drill down, you actually find out that that paddock's always outperformed the other paddock. Okay, and so there's a legacy of what was there before. So make sure you're comparing like with like. Make sure that what you're doing is repeatable. So I would encourage you to measure something now, use that as a benchmark, come back in a number of years and see if there's been a change in the direction that you might expect, because it's that change that you're really managing for. As with any sort of analysis exercise, we always submit our samples blind. We just number them 1 to 100, send them off, get the data back. I think that's a really important just general lab practice. And again, look for change. And I think above all, use soil biology as a filter for your management decisions. Okay? When you're doing something, think about what will be the impact of this practice on my soil biology. Okay, if that's all you get out of this is that it's just another filter that you look at your management through, then I think that's a really good outcome. So I will stop there, and I'm more than happy to take any questions you may have. Thank you.
Thank you, Tim. That gives us some insights in how
That's a really interesting question. So I would look at it almost the other way around, that what would be the effect of soil acidity on my soil health? What would be the effect of managing my soil acidity on my soil, like my soil biological health? And so I think what you'll see is a change through time, that if you try and ameliorate the acidity problem, you'll start to see improvements in the soil biology in your system. That will improve the resistance and the resilience and the buffering, and through time you should then see feedbacks into that process. But I think you need that first step of trying to address the acidity thing. That said, we're actually just setting up some experiments now with those omics techniques to try and look at the impacts of various things. We've looked at salinity, that has a massive impact on the microbial community. We're now just starting to look at effects of soil pH on that. What I would generally expect to see is that, and in fact in the meta-analysis there were different types of pasture in there. If you disturb the soil, you're coming through, you're breaking up the soil, you're breaking up that big hyphal network, and that reduces the ability, you know, if it gets all chopped up, it reduces the ability of it to form that association. If you add an excess of nutrients, the plant basically says, I don't need the fungus anymore, and so it reduces the reliance on that. If I can sort of move to an example in the vegetable systems that we worked in, what we found was that when plants were mycorrhizal, they didn't have any more phosphorus. The comparison was we had mycorrhizal and non-mycorrhizal plants with the same amount of nutrients added, and what we found was substantially more phosphorus in the plants that were mycorrhizal compared to where they weren't. And so the same might apply to pastures as well. That doesn't increase your yield necessarily, but it can impact your profitability. So if you can reduce the amount of input that you're having to add to the soil because you're getting your nutrients delivered through this biological pathway effectively for free, then that has an impact on the profitability. So I think often when we talk about mycorrhizas, people go, oh, mycorrhizal plant, non-mycorrhizal plant, no difference in yield. But if there's a difference in the nutrient content, maybe you could have got away with adding less nutrients, these external inputs that are so expensive. So again, specific examples, certainly annual pasture plants are mycorrhizal. They can be quite responsive to them in a positive way, and they will be impacted by the management, so tillage and nutrient addition, and maintaining cover and minimising fallow periods. Are the impacts on soil biology greater with, say, mechanical cultivation of the soil versus animal control of weeds?
I guess more specifically, nutrient losses.
Nutrient losses.
Yeah,
Yep.
by leaving the ground cover
Yep.
debris on the surface compared to putting it through an animal, is that more stability?
That's a really interesting question. So if you have an animal in the system and it's remaining in that system, it's going to consume the biomass, the plant biomass, it's going to excrete those nutrients back as urine and feces. They're going to cycle back through the soil process into the plants, and it'll keep cycling in quite a closed loop. So that's what nature does. It's a fairly closed system in that way. If that animal's leaving the farm gate, right, you're exporting those nutrients off-site potentially, so you could see that as a loss of nutrients as you export that through the farm gate. In terms of maintaining cover, again, the nutrients that are embedded within that biomass, they'll slowly get cycled through the soil process. The way I think about it is you're never going to stop the loss of nutrients through your system. What you want to do is minimize the leakage. Okay, so you want a non-leaky system, one that's less leaky, and the way that we reduce the leakiness of the system is you bring back in those biological cycles. So sure, the organic matter decomposes, it goes into mineral form in the soil, but it gets taken up by a plant and it just keeps cycling. Whereas if you're in a more disturbed system, you tend to lose those nutrients more rapidly. So I think anything that you can do to maintain the cycling of those nutrients on site will help with that.